The following video is a recording of a lecture from Genes to Galaxies, the 35th Professor Harry Messel International Science School, presented to high-achieving Year 11 and 12 students from across Australia and 10 other countries.
Okay, so what I will try to do this afternoon is quite different from what I did this morning. I will try to guess what will be the future state of biology. What are the present evolution of biological sciences? And so what I call the frontiers of limits of current biological research. Okay, it will be better. What are the frontiers? How to guess what will be the next results, the next developments in biology? You have three possible strategies. The first is to look at what biologists are presently doing. If you look at biological science, some research projects, some lines of research are very actively followed now. Many people work, for instance, on small RNAs. I will talk a little more about. Yeah?
Yes, great. I'm looking up to that.
It's good?
Yeah, it's good.
Okay, I got it. Okay, the first is to look, yes, it's better, the return. The first is to look at what biologists are doing. There are subjects which are now very actively, lines of research which are very actively pursued. So it's a first strategy to try to guess in which direction researchers are going. The second strategy to try to guess the future developments in biology is to look at what scientists consider as limits or things which have to be developed, research which have to be obtained for to go ahead in research. At every time in one science, if you ask scientists, they will tell you, this is a point we don't understand, we must work on this or this question. So it's a second strategy, to try to see what are the weaknesses in present knowledge. And it's particularly true when you consider the application of fundamental sciences. And in biology, many applications concern medicine. And if you look at medicine, it's obvious that some disease resists our present ways of acting. For instance, if you consider cancer, there were important progresses in the fight against cancer, but some forms of cancer are not cured now, and we have no real active drug against this cancer. So it means that probably we do not understand yet exactly what are the modifications of cells which occur during cancer. Consider other diseases like Alzheimer's disease or Parkinson's disease. In all these cases, there are descriptions of the disease, but there are no really drugs active against this disease. It means that probably our knowledge is still insufficient and has to be developed in the future. So these are the frontiers of knowledge, but I just want to warn you that probably what I will tell you will not what will happen in the future. And for the reasons which are shown here, François Jacob, who wrote these lines, was a Nobel Prize in Physiology or Medicine already 40 years ago. And in this book, published at the beginning of the 1980s, as you can see, he says that in fact, the future of science is always different from what we anticipated. And for the reason that a discovery cannot be a discovery if we know what we will discover. So if there are discoveries in science, it means that clearly there were things which were not expected. So what I will tell you is to try only to see these probably lines of research will be important in the near future, but I am clearly unable to tell you the biology of the middle of the 21st century will be that and that. Okay, it was a preliminary point, and I will now consider successively three different points. The first, I want to come back to what was maybe the most important transformation of biology during the 20th century, the rise of molecular biology, and to consider whether this important event is still important or whether we have to abandon this approach for a new approach. What I will try to show you is that in fact the molecular description are still very important. But probably what happens today is that they are complemented by other approaches, and I will try in the second part to describe these different new approaches. And finally, I will insist in the last part of this lecture on what I consider probably as very important for the evolution of biology. It's some of the encounter between evolutionary biology, we talked about this morning, and what is called functional biology. It means molecular biology, cell biology, biochemistry, and so on. And to tell you that in fact, these disciplines were quite distinct so far in the past, and now clearly more and more evolutionary biologists are interacting with functional biologists. And I think it's very important for the evolution of biology. So let's start to the first point. What is the role of what is the place of molecular biology now? So molecular biology, it was a description of biological facts at the molecular and macromolecular level. If one experiment is emblematic of molecular biology, it was the discovery by Watson and Crick in 1953 of the double helix structure of DNA. So description of molecules, of the structure of molecules, and understanding how these molecules interact to generate the complex biological phenomena. During the last years, many people have said that molecular biology was somehow dead. It was time to shift towards another approach of biological facts, considering a more global approach. Even some people say that finally, probably molecular biology was somehow a failure. It was not successful. It really focused too much on isolated molecules. It's high time to have a more global view of biological facts. What I want to show you is that I think it's not true. Molecular biology, molecular approaches, molecular description are still very important in biology. And to do that, I want to come back on what characterizes molecular biology. And I think what characterizes molecular biology is the idea that if you describe a structure, you will understand the function of the molecule and the role it plays. And the best example, in fact, is DNA. So you recognize on the right the molecule of DNA, and on the left, Watson and Crick presenting their models in 1953. And why this experiment was so important? Not an experiment, in fact, because just I open parentheses, but you know that, in fact, Watson and Crick did not do an experiment. The experiments were done by a woman, Rosalind Franklin, and they stole the result of Rosalind Franklin to build their models. One of the best examples of somehow misbehavior or misconduct in science. But nevertheless, they did a very important job by building this model. Why? Because when they built the model of DNA, from the model, as shown on the right part of the slide, it was possible to understand how the DNA molecule could generate two different copies of itself. So it explained why DNA was susceptible to be a genetic material. Because the molecule, the structure of the molecule, explains how the molecule can be directly copied into two identical molecules. The genetic role of the gene was explained by the structure of DNA. The structure of DNA also explains the origin of mutation, at least of some mutation. We talked about mutation this morning, but it explains why during the copying process, the replicating process, some nucleotides can be inserted at the wrong position, and for this reason you can create mutations, modification of the DNA molecule, and so mutations in the genes. And also, the molecule was at the origin of the idea of a genetic code. Because when you look at DNA molecule, it's quite monotonous as a molecule. And in fact, the only thing which can be significant is the succession of bases in the molecule of the different nucleotides. So it led Francis Crick to propose that, in fact, this succession of nucleotides is a code which explains how proteins are synthesized. It was the origin of the idea of a genetic code, the existence of which was demonstrated 8 years later. So you see the structure immediately opens to an understanding of the function. And this is characteristic of molecular biology. But it was not limited to DNA. For instance, when enzymes were described, the structure of enzyme, it explained how enzymes, proteins, are able to accelerate chemical reactions. And I just want to give you one other example of this power of structure to explain functions. And this example is shown here. It's what is called a potassium channel. It's slightly more difficult. I will explain a little, take some time to explain it. You know that in the nerve cells, you have what is called nerve influx. And it's a signal which allows nerve cells, for instance, to activate muscles or other nerve cells. This nerve influx is due to a propagation of change in transmembrane voltage along the nerve cell, along the axon of the nerve cell. This process is, in fact, due to the transient existence of currents across the membrane of the nerve cells. How are these currents generated? By the opening, transient opening of channels within the membrane of the nerve cells, which allows the passage of different ions. There is a channel for sodium, there is a channel for potassium, and some years ago, the structure of the potassium channel was determined by X-ray crystallography, analysis of crystal and diffraction of X-rays on these crystals. And why was this structure so important? It led to McKinnon, who was responsible for the structure, received a Nobel Prize 4 or 5 years after this obtention of the structure. It was important because, in fact, the structure, which is also represented here, you see the scheme represented the molecule within the membrane. This structure explains 3 different things. First, how is it possible for this channel to open when the membrane voltage is changed by the propagation of the nerve influx? So the channel is sensitive to the transmembrane voltage. How is it possible? This structure, with this structure, it was possible to provide an explanation of this sensitivity to the transmembrane voltage. Second, the channel close. It's a transient opening of the channel, which is necessary for the propagation of the nerve influx. And once again, with the structure, it was possible to explain why this opening of the channel was transient. I don't want to enter into the detail, but the opening of the channel is due to a movement of parts of the molecule called alpha helices. So change of voltage leads to a movement of these helices part of the protein, and it opens the channel. Closing of the channel is due to another movement within the protein, which puts one part of the protein on the top of the channel and encloses the channel. And finally, also, the structure allowed to explain why this channel was so specific for potassium ions, and, for instance, sodium ions are unable to cross the channel. I don't want to enter into the details of this last point. It's quite complex. But the important point, I think, is that with the structure, it was possible to understand the function of this channel and the mechanism of action of this channel. And this is really characteristic of molecular biology. So the question is, does this virtue of structure determination, this possibility from structures to explain function, is it disappearing? And my answer is clearly no. The determination of the structure is still very active in biology, very important, and leads to a lot of discoveries. And there are many reasons going in this direction. First, there are more and more powerful methods to determine 3-dimensional structures of protein and macromolecules. Just to give you one example of this technology, here you have the structure of a very big enzyme, which is polymerase, RNA polymerase, which is able to copy DNA to RNA during transcription process. You see the complexity of the structure, which was determined about 9 years ago, and also it led to another Nobel Prize. And why it was received the Nobel Prize for this work? Because it's highly important. When you have the structure, you understand many of the way this complex enzyme function. So methodology for efficient, more and more efficient technologies allowing to determine complex structures, and this complex structure explain a lot of biological facts. Second point, also is that these structures are not only important to understand what happens, but they are also very important to, for instance, design new drugs to eventually we can be used for to cure some disease. Just to give you one example, which is not so well known, but really this huge progress resulted from a knowledge of molecular structures. In the case of AIDS, you know that today there is an active therapy which is not perfect because it doesn't eliminate the virus, but it allows to people who have been infected to remain in good health, which is at least for a long number of years, which is something highly positive. And they call it free therapies because, in fact, you have a combination of 3 different drugs. In these 3 drugs, which are used together, there is one which was probably the most important. It's called antiprotease. It's an inhibitor of a protease, which is a protease of the virus. This protease, which is a protein which cuts other proteins, is important for the maturation of the virus. If you inhibit this protease, the virus is unable to mature and so to have its normal function. And this inhibitor of protease was obtained from the information which was obtained on the protease of the virus. So the protease of virus was produced in vitro, its structure was determined. From this structure, it was possible to design an active drug going at the active site and inhibiting the reaction. And this drug was tested, it was active, and now it's really the most important component of the therapy against HIV virus. So you see, this structural information is highly important. And today, for instance, in the case of cancer, you have now new drugs against cancer which are more specific of one form of cancer. And these new drugs have been designed from the knowledge of the genes or proteins which are activated during the formation of cancer. So once again, structure leads to a knowledge which can be used for obtention of new drugs. And finally, why is this molecular information still important? I think because if you consider organism, it's clear that what is encoded into organism is precisely the information to build macromolecules. So molecular biology was not one level of looking at one level of organization of organism chosen between many possible levels. I think it's a very important level. It's a level at which information is encoded within the genome. So I hope I have tried to convince you that this molecular approach of biological facts has still beautiful moments in front of it. So now I would like to shift to the second part. But new approaches can be combined with these traditional, not so long, but traditional because it already exists since 20, 30 years. New approaches can be added to this structural approach. And what kind of new approaches? The first of these new approaches can be simply the discovery of new phenomena. Consider, for instance, what happened during the recent years. Many people thought that the most important regulators, for instance, proteins which regulate the activity of genes, were proteins. It was the accepted dogma. And recently, it was discovered that small RNAs, or interfering RNAs, it's a technical term, are very important for regulation of gene expression. And if some of you are familiar, read some books or journals reporting recent results in biology, probably you have seen that work on this microRNA or interfering RNA are developing very rapidly. And even now, there are some medical application of this discovery because some small RNAs are used in some therapeutic approaches. So this was somehow unanticipated result. It's not, finally, it's not revolutionary. It's still the observation of molecule, characterization of the structure, but it's a new class of molecule, the importance of which was not anticipated. Another, I talked already this morning, that there are these phenomena of epigenetic regulation. So modification of DNA altering gene expression or modification of proteins surrounding DNA And there are many people working on this phenomena for the reason I mentioned this morning, their role in evolution, but also because they play a very important role in the activity of cells and organisms. Think, for instance, to a process like aging. Some people consider that maybe aging process might be due to some epigenetic modification of the genome. It's not admitted by all biologists, but nevertheless there are plenty of works done in this direction. Also in cancer, many people consider, that the main theory of cancer is that you have mutation, what is called somatic mutation in different cells, but many people consider that this mutation might be epimutation. So not replacement of one nucleotide by another, but modification of this type. So epigenetic phenomena are very actively studied now, and probably they will have a higher place in biological research in the next future. And also, last point, that in present approaches of biology, you have more and more what is called global approach. For instance, just consider what is called transcriptomics, the use of DNA chips. In one experiment, you can look at the level of expression of all the genes of one organism or one cell. And doing this kind of experiments in different conditions, you can look at connections between different genes, for instance, which are co-expressed in the same situation. From this, you can create, or at least observe, new links between processes which were so far separated in the description you had of cells. So this kind of new links related, for instance, proteins involved in cell division and proteins involved in the traffic of vesicles within cells. You discover this kind of new links. So now, thanks to the technology, it's possible to have a more global integrated vision of the function of cells. So this is not new, it's simply development of the molecular approach, but some of them were unanticipated. So now something maybe more new or a more important change in the traditional approach. There was during the last, let's say, 10 years, huge progresses, both progressing in physics, in apparatuses allowing the detection of very weak signals, and also progresses made in biology, with, for instance, the use of proteins which are called reporter proteins. These reporter proteins are proteins which are fluorescent, emit fluorescent light, and so it's possible to see this molecule within cells. So what is done by biologists is to take these proteins and to fuse them with the protein they want to study. And doing this, it's possible to follow the important proteins within cells. Maybe you heard that the last Nobel Prize this year in chemistry was given to researchers who have been able to develop these new technological tools. So, to be able to look at isolated protein, individual proteins. Another possible approach to look at individual proteins is to manipulate isolated protein, individual proteins, by very complex physical devices. I just want to give you one example of this kind of manipulation, some examples of this kind of manipulation and what they can bring to our knowledge. First example is the example of proteins like ATPase. So ATPase is a very important enzyme which is responsible for the production of ATP, which is the energetic molecule within cells. So production of ATP is fundamental. You have a complex structure, once again, the result of structural biology, structure determination. And this molecule, as you can see, have two parts. One part, which is in color here, is in fact turning rapidly, while the other part of the molecule is static. So ATPase is behaving like a small rotor, turning rapidly, like a rotor in the engine you have in machines. So this idea that you have a nanomachine, what is called a nanomachine, that ATPase is a nanomachine with this rapid movement, exactly similar to a rotor, was proposed some years ago. But how to demonstrate that? It's at a level where you can look at in a microscope because it's too small. In an electron microscope, as you know, it would be possible, but electron microscope, you can introduce only static objects. You can follow with an electron microscope movements. So how to look at this movement? What was done is shown on the next slide. what did the experimenters were to take the molecules and to fix on it a long chain of actin, which is another protein, but which formed very long chains. This actin, you can see, was fluorescently labeled. So thanks to this simple experiment, it was possible to observe with a light microscope the movement, this rotation movement of one part of the molecule relative to the other. Under the microscope, it was possible to follow the movement, and doing that, it was really possible to demonstrate that this protein functions like a rotor. So you see something which is not visible, but with some tricks, physical tricks, you are now able to see this movement, to amplify it, to see it under a microscope. Another example, it's now possible to detect unique molecules within cells. You see here on the left you have what was done. It was, as I mentioned previously, you have one protein, bacterial protein, and you put on this protein a reporter protein, a protein which is fluorescent. You join both of them, and with very sensitive physical devices, it's possible to see this individual molecule within the bacterial cell. You can see on the right you have two images. The images which is on the right shows the previous possibilities. You had light within bacterial cells, but as you can see, you have bacterial cells with some small sticks, but, well, you see only light, no precision. With new physical devices, it was possible to separate two spots, and each of these spots you see on this represent one unique protein. So you see that now it's possible with a light microscope to follow unique proteins, movement of unique proteins, the presence of unique proteins within living cells. And that's very important because it means that now you can follow at the molecular level what happens within a cell. For instance, you have a cell, you add an hormone, a factor in this cell, it will lead to a lot of transformation of this cell, a lot of events, and you can now follow directly what happens in the cell, what is the movement of the proteins, how a protein goes to the nucleus, and so on and so on. You can see it under the microscope with all these processes. So new possibilities to do a more dynamic description at the molecular level. And finally, doing that, you can also discover new phenomena, or at least show the existence of phenomena which were theoretically known, but so far not observed. And one of these phenomena is what is called molecular noise. Cells are small objects, and processes like transcription, for instance, is done by one protein, polymerase, which is acting very slowly for a chemist, at least, reactions are slow. And in this case, it was anticipated that the rate will not be constant. You have stochastic events, stochastic variation, due to the limited number of proteins and to the slowness of the reactions. What means, and this is called molecular noise. And for instance, what are the effects of molecular noise? Here you have yeast, which have, they are eukaryotes, so you have two copies of each gene. And with the kind of mechanism I described previously, the fusion of a protein coded by a gene with a reporter protein, it's possible to study the level of expression of, for instance, the two allelic forms of the same gene, the two copies of the gene. And when you do that, and you look at individual yeast cells, you can show that first, each yeast cell produces a protein at a different rate. The production is not regular, but you have production, non-production, production, non-production. It's stochastic. And it's stochastic even because if you compare the two genes, which code for the same protein, they are not expressed at the same rate. One will be expressed, it will be no longer expressed. The other is expressed, is no longer expressed. You have stochastic variation. It's not regular when you look precisely at within the cell. So you have noise. Cells are not functioning quite in a regular way. And it raises two questions, which is, if you have molecular noise, how did cells manage to somehow fight against this noise? Because if you have too much noise, it's clear that it will raise huge problems for cells. So probably this noise is controlled. And many researchers are now trying to understand how this molecular variation, these stochastic events, are controlled by cells. And the second is this molecular noise can lead to different states of the cells, because you have stochastic variation, so you have not exactly the same phenotype, as it means the same state of activity of your different cells. And many biologists consider that it can be important because it leads to, for instance, different states of activity of the different cells, and this variability can be important for cells, for instance, to adapt to new conditions. It creates diversity among cells of organisms which were identical at the beginning. So noise can be at the origin of a diversity which might be useful, but useful for what? That's the kind of question that researchers are looking at. So you see, in this part I wanted to show you that with this new technology, it's possible to discover new phenomena and to ask new questions within biological research. So now, other developments. So I shift to another part of these new developments. And it's a problem of modeling, doing models of complex biological facts. Here, maybe you are not familiar with this, this scheme represents the action of what is called a growth factor. Growth factors in organisms are very important. They are proteins produced by one cell, for instance, which will act on another cell to induce specific effects. In this case, you have one specific factor, which is what is called the vascular endothelial growth factor. This factor is responsible for the formation of blood vessels. So a factor which normally is not active in the organism. You don't create new blood vessels in a normal organism. But creation of new blood vessels can be important if you have been, for instance, injured, in case of you had a problem, or, for instance, in the case of tumors. When tumors grow, cancer cells grow, they create new blood vessels because they need oxygen, they need glucose. So cancer cells are able to induce the production of this factor, and it will lead to blood vessels which will somehow bring to the tumor oxygen, glucose, and other nutrients. So many biologists consider that looking at this, the way this factor is functioning is important, because, for instance, if it was possible to inhibit the action of this factor, it would be possible to prevent this formation of these blood vessels in tumor, and it would be a very efficient way to fight against cancer. There are also now some drugs developed from knowledge of this pathway. So you have this growth factor, you have this growth factor which binds to a receptor, and from this receptor you have a cascade of molecular interaction which will lead to the different effects of the factor within the cell. It's highly complex, as you can see on this scheme, because each small circle represents a different protein factor. So many complex interaction between them. If you look at such complex schemes and only look at, it's impossible to understand really what will happen. To really understand the consequence of the binding of the growth factor, you must have a more quantitative model. It means replace this qualitative description by a quantitative one with a question, be able to really look at the system and to estimate how it works quantitatively. But so to try to do a formal model of these complex relations. It's not an easy job to do. But to do a formal model is probably important to understand complex systems like this one, but it can be important also to understand more simple systems. I just want to give you one example, and you will understand immediately, I think, the interest of this modeling process. So modeling, it means a formal model representation of this kind of scheme. Here you have an example which is famous for many biologists. It was the first system of genetic regulation. So the first example of a mechanism of regulation of gene activity. It's what is called the lactose system. And so you have expression of 3 genes which are associated in the bacterial genome. And the control of expression of this gene is due to the presence of a repressor, a protein which binds to the 3 genes and prevents their copy into RNA. So regulation, negative regulation by a repressor. Okay, so if you now you consider how the systems work, so it's a system repressed by a repressor, but the repressor normally prevents the synthesis of enzymes which are necessary for the use of lactose, a sugar. But if lactose is present in the environment of the bacterium, lactose will bind to the repressor, it will inhibit the repressor, and in this case, the DNA, the gene will be transcribed, produce an RNA, and produce proteins which are necessary for the use of lactose, okay? So lactose is a sugar. If lactose is present in the medium, it will inhibit the repressor. Enzymes induced in lactose metabolism will be produced, and bacteria will be able to grow. But consider the system. In fact, the genes which are controlled, one of the genes controlled by lactose codes for a protein which is called lactose permease, which itself is required for the entry of lactose within the bacteria. So you have what is called a positive feedback loop. Why? Because if lactose enters into the cell, it will allow to produce the proteins which allow the entry of lactose into the cell. So you will have an amplification, a positive feedback. And with such a system, it was shown that this kind of system where you have positive feedback loops, mean a positive effect on the system and its functioning, it leads to what is called a bistable state of functioning for the system. It means that either the system is inactive, repressed, or it is fully active. Because once it has been activated, you have a positive loop stabilizing the active state. So you have two states, two extreme states, a bistable behavior. It was understood quite rapidly. 50 years ago, people had understood that it can lead to bistable effects. But the problem is that, first, it was impossible so far to demonstrate that such was the case. And second, it was also impossible to understand exactly why is the system functioning in a bistable way. Because you have other systems having the same structure with positive feedback loops, we do not really generate bistable states, two states of a different activity. Some of the systems with the same architecture leads to a progressive variation between inactive and active states. So explanations were not sufficient. And what was done in a study published some years ago was first to confirm at the level of bacteria that you have a bistable state of behavior, and second, to build a model and to try to understand with this model why you have such a behavior. So just look at the image which is at the center of the slide. This image represents bacteria, the expression of genes in bacteria, with the help of what I called previously reporter proteins. So you observe the population of bacteria, and in each bacteria, thanks to this reporter protein, you measure the level of expression. So in ordinates, you have the level of expression in different bacteria. So you see points, different points. In abscissa, you have the concentration of the inducer. So look at one concentration of inducer, you will see that you have two populations of bacteria. Either bacteria with a high signal or bacteria with a low signal. You can see, and you have no intermediate values. So experimentally, you have been able to clearly show that you have two states activity, bacteria which are fully expressing this gene, bacteria which are not expressing this gene, but you have not intermediate states. But now, thanks to this kind of observation, and they allowed to really determine the quantitative aspect of the expression, the authors build a model. So I don't want, this was the model they designed. They do a system of equations, and thanks to this model, they were able to now ask the question, why is the system bistable? What is fundamental to allow this bistable functioning, inactive, active? And they showed in particular that what was important was the affinity of the repressor for its site, for the DNA molecule. So once they have this information, it's a critical value of this affinity. If it's too low or too high, you will not have the phenomenon. Thanks to this information, what they did was to modify the affinity of the repressor by modifying the structure of the repressor, and they demonstrated that in this case he had not the bistable behavior. So you see that on a somehow simple system, thanks to modeling, it was really possible to understand why the system was functioning the way it was. And I think it's a good example to show that even in simple situations, mathematical modeling can bring answers to questions which had not been answered before. So you have plenty of works now done on modeling, and I think modeling, this kind of formal models, can be used for different purposes. First use can be to test hypotheses. Biologists observe facts, and so they do an hypothesis to explain the facts. Sometimes it's difficult to really, on biological systems, demonstrate that this hypothesis might be valid. So what is done now is to build models and to test with this model whether this hypothesis can be a good hypothesis to explain what is observed. And after, you must come back to the biological system and check that really this mechanism is operating. But I think it can be an important step. I want to give you one example once again. Okay, you see here is the cell membrane. You have a protein, the name is syntaxin, doesn't matter. But this protein, when biologists knew from previous studies that you can find this protein in the membrane either as isolated or as under the form of complex, as you can see on this side, either isolated molecule or aggregates of this molecule. So, how to explain this behavior? Why have you either isolated molecules or aggregates? The previous hypothesis was to tell probably there is something on the membrane. You have an heterogeneity of the membrane, for instance, of the lipid components of the membrane, so you have microdomains of the membrane, and it explains the different repartition of proteins. But in these studies, the people say, is it necessary to imagine a heterogeneity of the membrane of the lipids? We propose a simpler hypothesis. We propose that this kind of pattern of repartition of molecule can be explained simply by the existence of two strengths: an attraction between the isolated molecules and a repulsion when you have two bigger aggregates. Imagining only these two strengths at the molecular level, you can obtain this kind of repartition. But to do that, they did a model, quantitative model, and with this quantitative model, they were able to demonstrate that their model is susceptible to explain this repartition of this protein in the membrane. So the story is not ended. Now it will be necessary to really demonstrate that their hypothesis is good and that you have a strength of attraction, you have a strength of repulsion. But just to show you that models can be useful to validate, or at least some hypotheses, to show that one hypothesis is a good hypothesis and deserves to be tested more attentively. Just to, another possibility with models is to use models to show that a description has been completed, that now you have a good description. An example, I have no image for it, was the genes controlling early steps of development of Drosophila, so simple organisms. The genes are quite well known, and so it's possible to build a model of interaction, to do a formal model. And thanks to these models, people tried to reproduce what happened during early development. When they did that for the first time, the model was not working. It was impossible to reproduce what happened during early development. So they said probably something is missing. Our model is not containing all the components responsible for the regulation at this early step of development. So they come back to biological studies, genetic studies, and they showed that probably one additional protein, one additional gene was involved in the process. So they introduced this new component, they devised a new model, and in this case they were able to show that the model was properly describing what happened during early development. So you see that now a quantitative formal model can be used to check that you have reached a good description of your system at the molecular level. And finally, modeling is more and more important in one part of biology, one field of biology which is developing rapidly and which is called synthetic biology. I don't know whether you are familiar with synthetic biology. Synthetic biology is a part of biology where researchers try to modify previous organisms, but not like in genetic engineering by simply introducing one gene, but by introducing a fully a limited number of genes, but allowing a complex function to be realized. I'm slightly late, so I will take only one example of this. It was an experiment done some years ago called the repressilator. So the organism are bacteria. The system which is introduced is a molecular system which generates regular variations of activity. In fact, you know that we have all what is called an internal clock in our organism, which is a circadian clock. It explains why we suffer so much of jet lag. I can testify of it. But this circadian clock is controlling a lot of activities of our gene on a 24-hour period. So from this knowledge, and we know the molecular mechanism which generates this clock. From this knowledge, in this study, people generated an artificial system of proteins, but which was mimicking what exists in these circadian clocks, and which was susceptible to generate regular variation of activity. They introduced it into bacteria, and they obtained bacteria which are suffering from the jet lag, if one can tell that. It means that these bacteria are varying some activities regularly every 24 hours. What they did was to introduce a gene emitting light. So you see the bacteria every 24 hours emitting light. They become dark and they emit light 24 hours later and they come dark and so on and so on. So you have bacteria which now have a circadian rhythm, which is something not existing in nature. But this kind of studies, of work, would have been absolutely impossible without modeling, quantitative modeling, because if you directly try to build your system with different genes, it will not work. It was necessary to determine the rates of expression, the stability of proteins necessary for the system to work before constructing the system. So it's a characteristic that in synthetic biology, you want to introduce complex functional devices in organisms, and to do that, you need modeling. So we had two other examples of synthetic biology, but we will come back on the discussion if you have time. I now will shift on the last part of my talk, which is the present encounter between evolutionary and functional biology. I think it's a very important trend today in biological research, and somehow it will have a connection with what I said this morning. In 1961, Ernst Mayr, I mentioned his name this morning, which was a very important evolutionary biologist, remarked that in biology you have in fact two different kinds of biologists. You have evolutionary biologists and what he called functional biologists, molecular biologists, cell biologists. And these two kinds of biologists are doing different things, different studies, but they are answering also different questions. A functional biologist asks questions about how the cell is able to replicate, how such process work, how cells make DNA, make ATP, and so on and so on. So the question of functional biologists are how. The question raised by evolutionary biologists are why. Why such an organism has developed in such condition, why such variation was favorable in such environment, and so on and so on. And when you have a phenomenon in biology, in fact, you can ask two questions. There was one example given by Mayr, which was a problem of migration, bird migration. He took the example of the eastern coast of United States. In September, October, some birds migrate towards the south. And you can ask two questions. First is, how does the process occur? It means, what are the signals which lead to the migration? Probably change in temperature, changes in the length of the day. There are signals leading to different hormone production, leading to different behaviors and to the migration behavior. But you can ask also why questions. Why migration has been selected for? And the answer is probably because it allows the birds to find more food, to escape competition with other birds, and so on and so on. So one phenomena, two different kind of questions, two different approaches, evolutionary approaches and functional biology approaches. And in fact, these two approaches have no connection one with the other. If you are a functional biologist, you are satisfied by answering your question, how? And when you have answered the question, you stop there. If you are an evolutionary biologist, you answer why question, but you don't really are interested by the way this process were realized, by the how questions. And in addition, if you are an evolutionary biologist, many consider that in fact, natural selection selects for functions, not for particular structures. So probably to reach one function, you may have very different structures. So there was a separation between the two. But I think that things are now changing for some reasons that I have shown here. And to be brief, there are many now approaches in biology which require the two approaches to be combined. And the reason for these two approaches to be combined. The first is comparison of genomes. Now you have more and more sequences of DNA and of genomes. And once you have these sequences, what you do is to compare them. If you compare them, immediately you have access to an evolutionary history of the genome transformation, and so you shift from functional biology to evolutionary biology quite naturally. Second point I want to discuss is that in biology today you have what I call new researchers. In systems biology, synthetic biology, you have plenty of biologists which were in fact trained in physics, in mathematics, in other fields. And these new biologists somehow are not trained in this separation between the two forms of biology. So they naturally shift from question of functional biology to question of evolutionary biology. It's quite visible. They were not trained to consider that if you ask one kind of question, you must disregard the other kind of questions. Also, a point I have no time to develop, but which I think is very important, if you push a molecular description very far, and it's what has happened during the previous decades, naturally, a very precise molecular description leads to question about evolution of the system. If it's enough precise, the question of how the system evolved somehow springs naturally. And the last point is that it's possible now to do evolution in vitro, or as people say, to put the beagle in a bottle. It's a beautiful expression. It was in an article in Nature. It means that you have a population, for instance, of bacteria, and you can add, for instance, a new nutrient to which this bacteria was unable to metabolize, and you can observe how long it takes for this bacterium to be able to use this new substrate, new nutrient, and how the population of bacteria evolves by mutation, stochastic variations, and selection of the organism, the variation of which are favorable. So you can do limited evolution, but you can do evolution in vitro. And in addition, now with the tools you have to sequence very rapidly genome, it's possible to not only to observe the phenotype, how the bacteria change, but it's possible to look at what were the genetic mechanisms involved in the evolution of functions. So what were the mutations which occurred, what was the nature of this mutation, and questions like this. So for all these reasons, evolutionary biology and functional biology are now getting closer one to the other. And what will be the results of this encounter? So the results are, I think, multiple. And just, I mentioned this morning Evo-Devo, to try to relate evolution with developmental biology. It's a clear example of this encounter, because evolution is evolutionary biology, and developmental biology is a part of functional biology. So now the goal is to understand evolution by looking at the modification of specific families of genes. Also, with this new approach, it will be possible to explain the formation of complex macromolecules. Just to give you, I'm obliged to go faster. An example, you know that ribosomes are the structures which are responsible for protein synthesis within cells. It's very complex machineries, very complex structures. But the question which was raised in the article was, is it possible to imagine how this complex structure were progressively formed during evolution? Because it's impossible to admit that ribosome formed immediately as it is today. So probably there were simpler forms of ribosome. And the authors from structural studies tried to guess what was the progressive growth of the ribosome from a primitive form of ribosome to much more complex ribosome we know today. So to do an evolutionary history of macromolecules. And another example is here, complex structure of bacteria, bacterium, the flagell of bacteria. It's a very complex structure, you know, it allows bacteria to move. And in this study, once again, authors tried to guess how this complex structure had been progressively formed. What were the precursors of gene and protein which were the precursors of this complex structure? What was the evolutionary history of this complex structure? And I think this kind of study are very important in biology. I will tell you why I think it's particularly important. So it will be important also in the case of disease. And for instance, just to give you an example to understand disease, and in particular pandemia. Here we talk a lot about pandemia. Here it's what was called the Great Influenza. I don't know whether you know what was the Great Influenza. It was also named the Spanish fever. It was a pandemia which occurred at the end of the First World War. And more people were killed by the pandemia than by the war. It's not known from other people, but this pandemia, which is a pandemia of influenza, killed more people than the First World War. It was frightening because people were in good health in the morning and they were died in the evening. So very active virus. And how can you explain such a pandemia? And in fact, you have two different explanations. Explanation from functional biology. It was due to a virus with specific characteristics. And it's true. But you have also another explanation. The propagation of pandemia was due to the war and to special conditions existing during the war. This Spanish fever, which in fact originated in France, it's not fair to Spanish people, because, but this Spanish fever in fact originating among soldiers, because when you have armies, you have a collection of people together, so it's very favorable condition for the rise of pandemia. And it was the first event of the environment which explains the rapid development of the pandemia. And at the end of the war, all the soldiers came back to their countries and they propagated with them the pandemia. So this movement of people, in fact, was a movement of the virus. And it explained why the pandemia started in 1917, just before the end of the war, but the worst year was 1980 and 1919. Because the soldier brought with them the virus. So you see, to explain the pandemia, it's probably the same for the swine fever pandemia we know today. You have both to look at the virus, functional biology, and to look at the propagation of a virus, which is a form of evolution, in fast evolution of the virus. So, and last point is why is it important to bring together both knowledge, functional biology and evolutionary biology, because I think it's the only way to have less naive vision of what is evolution. And just to take one example, which is, I think, maybe most important, at least as far as we are concerned, it's human evolution. Many people now, from the knowledge of human genome, monkey genome, try to understand what were the specific genetic events at the origin of the modern human beings. If you leave functional biologists alone, they will provide very naive scenarios of what happened. And I will skip the first example, but just focus on the second example. The example of the language gene. Maybe you heard about that. It was a gene which was discovered because some mutation of this gene lead to some problem in speech. The individual who would suffer from this mutation have difficulties to organize their speech. They don't respect the rules of construction of sentences. So it was discovered in one family, and the gene. Huh? What happened? I did nothing.
Okay. That's not good. Okay.
Okay.
It's back up here. Okay, 1 moment. Okay, we'll be ready in 90 seconds. We'll be ready in 90 seconds.
Okay.
It's a bit...
I made the mistake? No?
Oh no, I think
It's not.
the system just crashed.
It's the second screen also.
Yeah, it'll come soon enough.
Okay. I can continue. We will see the image after. So this gene was characterized. This gene whose variation is responsible for some problems in speech. And its structure was determined, and the structure of the gene was compared with the structure of the gene in monkeys and other animals. And what was discovered was that this gene had been active, had been stable in most mammals. So a very stable gene. The structure has not been modified during evolution of mammals, but surprisingly, in the human lineage, there were 2 mutations in the gene. So the conclusion of the authors was, so we have the solution of human evolution. This gene was changed during human evolution. It allowed the formation of the language. And so that's the reason why we spoke and the reason why human beings are what they are.
Okay, almost there.
Now I need the image for 3, 2, 1. And nothing happens. It works, it works. Okay, so it was a family I described previously. This was the comparison of genes with that you can see in the upper part, these 2 mutations which specifically occurred in the human lineage. Hence, you had the hypothesis which was proposed, which is summarized here. So, you see, you have a pig. He received the gene transplant, so he received this gene, and is now able to speak. You introduce the gene, and it's as simple as that. And so now we ask what we will eat for dinner. Not me, I hope. So, these are very naive vision of evolution, but and I think it's clearly this naive vision are coming from functional biologists. We consider that you have 1 gene, 1 function, and it's sufficient to explain evolution. So, putting together the work of evolutionary biologists and the work of functional biologists will allow this scenario to be far less naive. And so, I'm sorry, I have no time. And we come back to a point which was raised during discussion. I think also it's this convergent movement of evolutionary biology and functional biology will be good to fight against intelligent design, supporters of intelligent design. Because if you look at the arguments which are used by people supporting intelligent design, so the idea that science will never be able to explain, for instance, the formation of organisms, many of the arguments are in fact arguments which come from biochemistry and molecular biology, telling how can you explain the formation of complex metabolism, how can you explain the formation of a bacterial flagellum. It's too complex, it's not possible to explain it naturally. So I think if really biologists work on this question and are able to provide scenarios, it will be the best weapon against intelligent design. And finally, as a general conclusion, I think maybe 2 more changes very rapidly. First, biology during the 20th century was characterized by the use of animal models. I will stop 1 minute. And animal models like bacteria, bacteriophage, Drosophila. What is new in biology now is that many more models are used. And I think it's thanks to fast genome sequences. So now there is not 1 unique model to study a biological process, but more and more models. So biology is more open to diversity. And second point I want to raise, and it will be the last, when molecular biologists discovered DNA genetic code, many of them considered that they have discovered the secret of life. And it was what Crick, for instance, said. After many people considered it was a too naive vision. But I think there was something true in this. It means that the main rules of governing the functioning of organisms has really been discovered. But what has not been discovered is the way these complex functions were progressively put together within one unique object, the first cell. So the question of life is no longer a question of function, it's a question of history, how the process naturally happened, how the first cell was formed. So, okay, I was slightly too long, sorry.
Thank you.